DNA origami, creating tunable Moiré superlattices for nanotechnology.
Imagine a world where scientists can design and build materials at the nanoscale with unprecedented precision and control.
Sounds like science fiction, right?
But here's where it gets real. Researchers have successfully created DNA Moiré superlattices, periodic structures assembled from DNA origami, that can be tuned to have specific properties and applications.
In a groundbreaking study, scientists have demonstrated the ability to create complex, periodic structures using DNA origami, a technique that involves folding DNA molecules into specific shapes and patterns.
By carefully designing the building blocks and using seed components to direct assembly, the researchers were able to create DNA Moiré superlattices with long-range order and tunable properties. But here's where it gets interesting. The periodicity of the Moiré pattern can be adjusted by changing the size difference between the DNA origami components. This means that scientists can essentially dial in the properties they want the material to have, opening up a whole new world of possibilities for applications in fields like metamaterials, nanophotonics, and nanoscale templating. The researchers used a combination of advanced techniques, including atomic force microscopy, dynamic light scattering, and fast Fourier transform analysis, to characterize the assembled structures and understand the underlying assembly mechanisms.
They also used molecular dynamics simulations to optimize the design of the DNA origami components. And this is the part most people miss: the seeds play a crucial role in directing the nucleation and growth of the superlattices. By carefully controlling the size and arrangement of these seeds, scientists can essentially program the material to have specific properties and structures. The potential applications of these DNA Moiré superlattices are vast and varied. They could be used to create metamaterials with unique optical and mechanical properties, design nanophotonic devices with tailored light matter interactions, and provide nanoscale templates for the fabrication of other nanomaterials. Furthermore, the structures could contribute to the development of biomimetic materials inspired by natural structures. But what does this mean for the future of materials science? Could we see a new era of designer materials that can be tailored to have specific properties and applications? The possibilities are endless, and this research is just the beginning. As we continue to explore the possibilities of DNA nanotechnology, we are left with more questions than answers. Can we use this technique to create materials with unprecedented properties and applications? How will this impact our understanding of the natural world and our ability to design and engineer materials at the nanoscale? The future is uncertain, but one thing is clear: the potential for DNA nanotechnology is vast and exciting. Revolutionizing light and sound, DNA Moiré superlattices explained. These DNA structures could transform our understanding of light, sound, and matter. In an extraordinary advancement, researchers have successfully engineered DNA to function as a molecular architect, crafting complex Moiré superlattices with an unprecedented level of precision. By directly programming twist angles and geometric configurations into DNA strands, scientists have developed self-assembling nanostructures that have the potential to revolutionize various fields, from optics to quantum materials. These DNA-constructed lattices autonomously form in solution, eliminating the need for manual stacking. This breakthrough paves the way for materials capable of directing light, filtering spin, or vibrating in finely tuned manners, all at the nanoscale. Revolutionizing Moiré superlattices with DNA-based design. Moiré superlattices have emerged as a focal point in advanced research within condensed matter physics and photonics. Traditionally, constructing these structures has been a labor-intensive endeavor, requiring meticulous layer alignment and transfer techniques conducted under strictly controlled laboratory conditions. Our method circumvents the conventional limitations associated with creating Moiré superlattices, states Professor Laura Nalu, who leads the Second Physics Institute at the University of Stuttgart. A self-assembling framework for nanoscale structures. In contrast to traditional techniques that depend on mechanical stacking and twisting of two-dimensional materials, our approach utilizes a bottom-up assembly process, explains Laura Nalu. This assembly process involves linking individual DNA strands to create larger, organized structures. It is guided by self-organization, meaning the DNA strands connect independently through molecular interactions, without external assistance. The Stuttgart team has centered their methodology around this principle. We encode the geometric characteristics of the superlattice, such as rotation angle, sublattice spacing,
and lattice symmetry directly into the molecular design of the initial structure, referred to as the nucleation seed.
We then allow the entire architecture to self-assemble with nanometer accuracy.
This nucleation seed serves as a molecular instruction manual, directing the growth of two-dimensional DNA lattices into precisely twisted bilayers or trilayers. Remarkably, this entire process occurs in a single step within a liquid solution, negating the need for intricate manipulation.
Bridging the gap in the intermediate nanometer realm.
While Moiré superlattices have been extensively studied at atomic, angstrom, and photonic, submicron, scales, the intermediate nanometer range, where molecular programmability and material functionality intersect, has largely remained unexplored.
The Stuttgart researchers have successfully addressed this gap in their latest study. The team integrates two powerful DNA nanotechniques, DNA origami and single-stranded tile (SST) assembly.
Employing this hybrid approach, the researchers constructed micrometer-scale superlattices with unit cell dimensions as small as 2.2 nanometers, featuring adjustable twist angles in various lattice symmetries, including square, kagome, and honeycomb. They also showcased gradient Moiré superlattices, where the twist angle and, consequently, the Moiré periodicity varies continuously throughout the structure. These superlattices exhibit well-defined Moiré patterns under transmission electron microscopy, with observed twist angles closely aligning with those encoded in the DNA origami seed, notes co-author Professor Peter A. van Aken from the Max Planck Institute for Solid-State Research. The study also presents a novel growth process for Moiré superlattices. This process begins with spatially defined capture strands on the DNA seed that function as molecular hooks to accurately bind SSTs and guide their interlayer alignment. This enables the controlled creation of twisted bilayers or trilayers with precisely aligned SST sublattices.
Exciting applications from optics to spintronics.
The high spatial resolution, precise addressability, and programmable symmetry of these new Moiré superlattices offer significant potential for a wide range of applications in both research and technology. For instance, they serve as ideal scaffolds for nanoscale components such as fluorescent molecules, metallic nanoparticles, or semiconductors arranged in customized 2D and 3D architectures.
When chemically transformed into rigid frameworks, these lattices could be repurposed as phononic crystals or mechanical metamaterials with adjustable vibrational responses. Their spatial gradient design also opens new pathways for transformation optics and gradient index photonic devices, where Moiré periodicity could guide light or sound along controlled paths. One particularly promising application involves spin-selective electron transport. DNA has demonstrated its ability to function as a spin filter, and these well-ordered superlattices with defined Moiré symmetries could provide a platform for investigating topological spin transport phenomena in a highly programmable environment. This is not merely about replicating quantum materials, asserts Laurent Alou. It's about broadening the design landscape and enabling the construction of new types of structured matter from the ground up, with geometric control inherently integrated into the molecules. Reference: DNA Moiré superlattices by Xinxin Jing, Nicholas Kronberg, Andreas Piel, Benjamin Renz, Longjiang Ding, Tobias Heil, Katharina Hipp, Peter A. van Aken, Hao Yan, and Nalu, July 17, 2025, Nature Nanotechnology. Never miss a breakthrough. Join the SciTech Daily newsletter. DNA Moiré superlattices, programmable nanostructures with tunable properties. Prepare to have your understanding of nanotechnology challenged. Scientists have just created something that was previously thought nearly impossible: programmable DNA Moiré superlattices at the nanometer scale. But here's the twist: this breakthrough fills a long-standing gap between atomic and microscale Moiré engineering, introducing a controllable, bottom-up approach to a notoriously tricky length scale. Interested in how bending and stacking DNA, yes, the stuff your genes are made of, can reveal new physical phenomena? Keep reading, and let's unravel what might be one of the most provocative advances in self-assembling materials. Let's start with the basics. Moiré patterns are fascinating geometric effects found at all scales, from everyday fabric weaves to atomic layers in advanced materials. Picture two slightly mismatched grids placed atop each other. Differences in their spacings or a small twist can produce a new, larger repeating pattern called a Moiré superlattice. Legendary in physics, the Moiré phenomenon has been harnessed in materials like magic angle twisted bilayer graphene, where aligning two graphene sheets at 1.1 degrees not only creates a superlattice 12.8 nanometers wide but fundamentally alters electronic structure, unlocking exotic quantum states.
This has even ignited a new field called twistronics, which is revolutionizing how scientists think about electronic and photonic devices. But here's where it gets controversial. These advances have been mainly restricted to two realms: angstrom scale, atomic, and submicrometer scale, artificial materials. At the atomic scale, it's relatively straightforward to access suitable lattices, think van der Waals materials, while photonic metasurfaces are within reach for the micron scale, thanks to modern lithography. But when it comes to the nanometer range, roughly several nanometers in lattice spacing, there's a frustrating barrier. Natural materials and standard fabrication techniques just don't cut it.
The result? A yawning gap right in the middle of materials science, ripe for innovation. This is where DNA nanotechnology boldly enters the scene.
Using ingenious bottom-up design, researchers constructed entirely new classes of Moiré superlattices from DNA, bridging that daunting nanometer gap.
How?
By engineering programmable DNA origami seeds, tiny, twisted DNA bundles that act as templates, and using controlled self-assembly of single-stranded DNA tiles, SSTs. These nanoseeds aren't simply passive scaffolds. Their internal twist and modular binding sites actively dictate the stacking, rotation, and registration of 2D DNA sublattices around them. Now, let's clarify for beginners. DNA origami is a technique where long strands of DNA, combined with many short staple strands, fold into specific 2D or 3D shapes, much like paper origami, only billions of times smaller. In this work, engineers created two types of hollow DNA bundles, called seed S and seed H, with precisely controlled geometries and internal twist angles. These seeds feature distinct regions for starting and growing DNA layers, but crucially, they include special segments like Z2 and Z4 that precisely set the twist between layers, controlling the resulting Moiré pattern. And this is the part most people miss. The twist angle imposed by the seed doesn't necessarily match the overall helical twist of the seed itself, only the dedicated segments, Z2/Z4, matter. For example, one seed variant induces a 3.8 degrees right-handed twist per stack layer, another configures a subtler 1.2 degrees twist. This remarkable modular control is verified by transmission electron microscopy, TEM, confirming that the nanoseed structures look just as designed.
As for the 2D DNA sublattices? By orchestrating DNA tile sequences and assembly conditions, the team built square, kagome, and honeycomb lattices, each with their hallmark symmetries. Intriguingly, the kagome lattice emerges by relieving strain within the square lattice, acting as a loosely packed square with characteristic voids. To the naked eye via microscopy, these lattices are sometimes distorted round, rhomboid, or irregular hexagons, reflecting the stresses of dense DNA packing, but at their core, their atomic scale regularity shines through, especially in ultra-high resolution, cryo-EM images. But achieving these intricate Moiré lattices isn't just about making the right building blocks. It turns out that seed design and nucleation pathways are everything. In unseeded DNA assembly, new lattices grow slowly, as individual DNA tiles randomly bind and nucleate. Seeding, on the other hand, accelerates and directs the assembly dramatically, but only if the seed presents the correct pattern of capture sites. These captures act as molecular glue for two neighboring domains on a DNA tile, and their arrangement, fully paired, partially paired, or alone, switches the system into fully cooperative, partially cooperative, or non-cooperative nucleation regimes. Why does this matter? Imagine the challenge of painting a wall. If the first coat of paint sticks perfectly everywhere, the second layer goes on smooth. But if you have missing patches, the second layer will be full of defects. Similarly, only seeds with complete, continuous pairs of capture sites lead to robust, uniform bilayer assembly. Split up or scatter the capture pairs, and you get patchy, random growth. Remove them entirely, and you're basically back to square one, no better than letting the DNA tiles assemble willy-nilly. This principle was confirmed not just with square lattices but with kagome and honeycomb variants too, demonstrating a fundamental, symmetry-independent law of successful DNA-based Moiré superlattice assembly. Here's a surprising twist, pun intended. The size and quality of the resulting superlattices depend not only on these molecular details but also on concentration and temperature. Want mostly bilayers and fewer single layers? Use more seeds, but be aware, higher seed amounts nucleate more sites, yielding smaller superlattice domains overall. Lower temperatures mean homogeneous nucleation creeps back in, causing higher variability. Robust assembly, it seems, walks a fine line between temperature, seed design, and DNA concentration. The researchers didn't stop at bilayers. Armed with their modular framework, they constructed trilayers, hybrid superlattices, mixing, for example, square and kagome, and even gradient Moiré structures. These latter ones stand out. By employing an asymmetric, trapezoidal DNA seed, they induced a gradual shift in lattice orientation across a DNA layer, creating a gradient in Moiré periodicity, a feat rarely observed at this scale. If you think about a rainbow's gentle change in color across the sky, that's what's happening here at the nanometer level, but with DNA tiles instead of light. Does this approach upend traditional epitaxy? Consider this controversial comparison. In standard crystal growth, surfaces or lattices serve as templates, directly guiding new material to stack up in registry with the underlying crystal. But DNA Moiré,